Okay, so I'm going to take you through our cell cycle standard, and to be proficient, you need to interpret a model and decipher that model. So looking at pictures of different stages of the cell cycle, you need to be able to put them in order. You need to name those phases and give characteristics of each phase, at least one characteristic of each phase. So let's get started. These are basic pictures we're looking at. And this is a process where one cell becomes two cells. And what we start with is interphase. And an interphase, I call it kind of the spaghetti time because the DNA isn't coiled up, it's loose. And it's just this loose chromatin where it's easy to copy it, but it's also easy to read it in order to make proteins. So interphase has three stages or phases within it. The G1 phase where the cell grows, the S phase where the DNA is copied, and the G2 phase where the cell continues to grow and continues to do protein synthesis. So again, even though this looks like a small portion of the overall process, time-wise, it's by far the longest amount of time that the cell spends. And again, like thinking about this whole process is happening 3 million times a minute within you. So you're making 3 million new cells every minute. Pretty incredible. After interphase comes prophase, and in this phase, I kind of talk about the spaghetti turning into sausages because it's much easier to, like, if you had a plate to spread sausages, half of the sausages on one side, half on the other side. Whereas with the spaghetti, it'd be hard to pull out all these loose strings that are kind of interwoven with each other. So again, prophase, I think of it as the sausage phase, where we begin to get the chromosomes to appear and coil up. The nuclear membrane breaks down. You can't see that in the picture, and you also can't see in this picture the centrioles migrating and then beginning to send out spindle fibers, but that also is in prophase. Sometimes it's called prometaphase, but we're just going to call it prophase. After prophase comes metaphase, and meta we say middle. Okay, so what does the middle stand for? It means that those chromosomes are lining up in the middle of the cell, and these little ropes or lassos from the that are spindle fibers, which are sent from the centrioles, are going to grab onto each of the chromosomes that have been duplicated. And we call those sister chromatids, but if you keep calling them chromosomes, that's okay. Anaphase comes next, and ana is a part, so pro meta ana tella, and in anaphase, ana is a part. So in anaphase, the chromosomes, you can see in the diagram there, it shows them kind of pulling apart towards each end of the cell. Telophase is 2, so tella is 2. And what that means, you can see that dotted line surrounding the outside of the chromosomes here. So in telophase, we have these nuclear membranes, and again, the nuclear membrane is what surrounds the DNA. The nuclear membrane is reforming. The two nuclear membranes form back around the chromosomes. So it's still one cell, but we're getting close to being able to divide the entire cell. And when we actually do divide the cell, cytokinesis is that final stage, and it's split. So the cell is officially split into two cells. And now you can see they're small, right? So this process is going to loop back around to the G1 phase where it grows and starts becoming this size again. Maybe you've heard of the term mitosis. Mitosis is division of the nucleus. So those four middle stages of this is mitosis. And we start with one nucleus and we end up dividing it into two nuclei. And then the entire thing, though, is called the cell cycle. So there's different ways we can try and remember all this stuff. A lot of it's just repetition. Prophase, metaphase, anaphase, telophase. With prophase, I often say CNCS. And then meta is middle, ana is a part, tella is 2. Okay, G1S, G2. It grows, replicate, grow. Replicate means copy, where the DNA is copied. And it's not copying or replicating the cell, it's just copying the DNA. All right, so some practice here. On your test, they're going to be scrambled, and you got to try and pick the order and name those, and then give a characteristic of each. So if you look at these pictures, which one is going to go first? And we start with spaghetti, and that's interphase, and that is this one. From spaghetti, we go to sausages, which is prophase, which is this one. Then they got to line up in the middle, which is metaphase, PMAT, prophase, metaphase, anaphase, telophase. Metaphase is middle. We line them up in the middle. And then we need to pull them apart. So then we have anaphase, and then we have two nuclei, which is right there. And then from there, we split them. So the cell splits in half, and then we can start back at interphase. Again, part of this, you have to put them in order, so it would be F, E, B, D, C, A in this case. Okay, one more example. Again, what do we start with? We start with interphase, and that's the spaghetti phase, so that's there. The cell is growing. This is the normal time that the cell is just basic life going on here. It's growing, it's making proteins, carrying out life functions. But if it goes to the S phase, then we copy the DNA, and the cell grows a little bit more prior to going into prophase, which is our sausage phase. Okay, the chromosomes coil up. This picture doesn't demonstrate it, but we also lose the nuclear membrane. The centrioles are going to go towards the poles and start sending out spindle fibers. So that brings us to metaphase. And again, the centrioles are really small. You can't really see them here, but where these spindle fibers are coming from, those would be the centrioles on each side. Where that's what we say migrate. They're migrating to the edge as opposed to just kind of being located close to the nucleus. So meta is middle. Ana is next. Ana is apart. So which one shows that they're apart? That one. Chromosomes are pulled apart. And then tela is two nuclei, which is that one. And then finally cytokinesis, and they split. Okay, so again, if you look at it in a chart, we've got interphase, we've got mitosis, and we've got cytokinesis. Interphase is G1, SG2. Mitosis is PMAT, prophase, metaphase, anaphase, telophase. G1 is grow, S is replicate, G2 is grow again. Prophase, CNCS, the chromosomes appear, the nuclear membrane goes away, the centrioles migrate, and the spindle fibers are sent out. Metaph is middle, so the chromosomes are lining up in the middle. Anaphase, they're pulled apart. Telophase, we get two nuclei existing in one cell as they form back around the chromosomes. Then cytokinesis, we split the cell. If we look at it in another way with a slightly different picture, here are centriole pairs. Here is all of that spaghetti-looking chromatin, the DNA material. In prophase, you can see we're losing the membrane and these long strands of spaghetti. Again, they've been duplicated, so why are there two strands next to each other? Because, like for example, with humans, we have 23 pairs of homologous chromosomes. That's ultimately 46 strands of spaghetti. Those 46 strands get duplicated, so before we actually head into this phase, we've got 92 individual lines of spaghetti. They're just 46 paired up here. Anyways, these centrioles are starting to migrate. They're starting to send out these spindle fibers. Metaphase, all of these spindle fibers are attached to each end of these chromosomes, these sister chromatids. In this phase, there's a little checkpoint to make sure everything's attached correctly. Otherwise, we can end up with non-disjunction, where we pull too many chromosomes to one side and not enough to another side. But meta is middle. Anaphase, where they're attached, we start pulling them apart. So we're starting to separate the DNA to two sides of the cell. Telophase, you can kind of see here, we're starting to reform a membrane. around the individual sets of DNA. And finally, I can see this like cleavage here where it's starting to form in, but it doesn't officially split until cytokinesis. We have two cells and we're back to G1 phase where we're in spaghetti time. Okay, and part of mitosis is that we end up with identical copies of the initial. Meiosis, we do 2 cycles of this and we end up with half as much DNA. Okay, so again on your test, you get a random order. You have to put them in the correct order and name them, and you also need to give characteristics of each phase. And that's it. Hopefully that's helpful for the cell cycle.